For example, just to finish, one example of how can we use networks to understand very complex information. So this is one example of one study that has been performed by our group in plants under water density in red and well watered in blue. So looking to the heat map, so what kind of information you can have from this heat map? It's pretty difficult to have, right? So you have here, each line of this heat map is a metabolite from the top down. You have serine, alanine, butyric acid, pyroglutamate, glutamate, and so on. So you have many metabolites, and here is the number of metabolites not that high. You can have, for example, in terms of transcriptomics, you can have more than 20,000 genes. How we are going to interpret this? Look into the heat map. Just look into the heat map. So it's pretty difficult, especially in this case that you don't have a clear difference. But what we can do is using some systems approach. One of them is the multivariate analysis. For example, in the right side you have a graph of ortho-PLSDA that you can have. You summarize all of this information that you have in the heat map in a single graph, and you see if the treatment of water deficit has changed the plant at metabolic level. Okay, so take a look in the first. These samples are not clearly, but they are separated by the first component of the axis x. So you have here imagine a line from 0 here that is splitting the water depth samples to the left and the well water samples to the right. So I think it's kind of clear that you have a separation of this information. What this graph is saying, so it's taking all of this information and it's providing to your answer. Yes, water depth condition has changed the metabolism of these plants. But still, we don't know, with only this information, we don't know why. And then you can go to the networks. What we are going to do in the next class is building some of these networks. For example, this is a network created by Cytoscape, which is the software that we are going to use. What kind of information you have here? It's a pretty complex network. You have null compounds, unknown compounds. The correlation strength between the metabolites is given by the thicker arrows. You mean have more the metabolites that have more have higher correlations between them. Positive correlation, you see red links, and negative correlations, you have blue links. Okay, so here it's how the network is created, but then you can, and we are going to do that, we can try to bring some information from this network. For example, here are the networks of this study. So the software allows you to build the network in different ways. So this is the same network as the previous slide, but in a circular mode. So you can, here I think it's even clear maybe that the red network is more connected. You have more links. This is what I'm saying here. So you have more links, you have more network, more connected networks. And this is becoming something, we are seeing this in many works. It's not always, but under stress conditions, plants has more connected networks. So this seems to be almost a pattern of response, but some cases we don't see this, but in most cases we see this. Under stress, plants has more connected metabolic networks. Another example from the work, another work from our lab, we challenge the plants against including virus in the leaves. So this is the metabolic network of plants without virus. So you see that you have some connections. So connections here are significant. And if I'm not wrong, the red connections are positive and the blue connections are negative. Yeah, that's it. And the thicker the link, higher is the Pearson correlation. So you have different modules here. For example, succinate, butyrate, valine, and isoleucine, they are parts of the network. They are isolated. Okay, here you have different, so 1, 2, 3, 4, 5, 6, 7, 8, and 8 modules, and inositol, glycolate, and glycine are not connected to any other metabolite. So this is a very simple network, right? So malate is a hub here, proline is a hub here, but the characteristics of this network of copy plants under non-stress is that you have different fragments. The network is fragmented. But what happened when we have virus? So the virus changed everything in terms of network. So instead of have different, instead of have several fragments of connections between metabolites, I mean subnetworks, we have one huge network here. So many metabolites are connected to each other. You have aspartate and perhaps the citrate and sucrose are the main hubs here. We have another module which is pyruvate, serine, and succinate, and we have an interesting module here. They are highly connected to each other, which is leucine, isoleucine, homoserine, and valine. So this module here, this subnetwork composed of these 4 metabolites, brings our attention. And actually, we have made many other analyses, and we are going to go deeply to investigate. Perhaps this plant is resistant to the virus. So the virus cannot, can affect, but the plant is more resistant than others. So we want to understand why. And based on this analysis and also other analyses, we are going to go deeply on the genes and proteins of this metabolic pathway here, valine, isoleucine, leucine, and homoserine, especially leucine and isoleucine and valine. So we are going to try to manipulate these genes and incorporate in a plant that is sensitive to this virus. Perhaps we can create a resistant cultivar that cannot survive under virus attack.